Did I say humans? Yes. Enough gross fly talk for a moment. On to slightly less gross things. Problems that occur when chromosomes or chromosome numbers are altered. It happens, unfortunately, more often than we politely talk about. Miscarriages or spontaneously aborted pregnancies often result when very large errors occur, and many viable disorders do also exist. Plants tend to be more robust to large genetic changes than animals do, as we shall see. Meiosis, like mitosis, is a fantastically efficient but not perfect cellular process. Sometimes the microtubular apparatus makes mistakes. The main type of error is called non-disjunction. In non-disjunction, the microtubules fail to connect properly, and so one daughter cell gets too many chromosomes and another one gets too few. So here's what that looks like. In meiosis 1, the homologous chromosomes line up at the metaphase plate, and normally they all separate. In non-disjunction, the homologs fail to disconnect, and both daughter cells end up with the wrong number of chromosomes. Non-disjunction can also occur in meiosis 2, where the sister chromatids fail to disconnect properly. Whether the non-disjunction occurs in meiosis 1 or meiosis 2, the result is at least two gametes with the wrong complement of chromosomes. Cells that have the incorrect number of chromosomes are called aneuploid. Ploidy, as in haploid or diploid, meaning the number of chromosomes. EU, means true or correct, and the prefix an, means not, so aneuploid means not the correct number of chromosomes. When a zygote has one missing chromosome, we call that monosomy. When there's an extra chromosome, 3 instead of 2, it's called trisomy. In this karyotype below here, you should note there are no aneuploidies. There are 2 copies of each chromosome. Sometimes entire sets of chromosomes are copied, and there can be an entirely extra set of chromosomes. This leads to multiple sets of chromosomes, and it's called polyploidy. Triploid means there's 3 sets of chromosomes. Tetraploid means 4 complete sets. This phenomenon is very, very common in plants, to the point where plant breeders try to produce polyploid plants. Most of our varieties of wheat that we eat are hexaploid, with 6 sets of chromosomes. And the strawberries we find in the grocery store, which are much, much larger than the ones you might encounter in nature, they are that way because they are octaploid, or have 8 sets of chromosomes. Having complete extra sets of chromosomes is much more tolerable than being aneuploid. Remember this plant from chapter 12? The adder's tongue fern? Our champion of the chromosomes is undoubtedly the result of probably several rounds of polyploidy. There are a few viable aneuploid conditions that occur in humans. One is Down syndrome. People with Down syndrome have 3 copies of chromosome number 21, so another name for this condition is trisomy 21. It affects less than 1% of children born in the U.S., though older mothers are more likely to give birth to babies with Down syndrome for reasons that are still not well understood. You can see in the karyotype here that this young girl has an extra copy of chromosome 21. The symptoms of Down syndrome include physical and intellectual delays, and the eyes have a familiar shape to them, which is called an epicanthal fold. In addition to trisomy 21, humans may survive to term with a few other aneuploid conditions, almost all of them to do with the sex chromosomes. Klinefelter syndrome, or XXY, is when a male is born with an extra X chromosome. Klinefelter males, humans anyway, often don't express many symptoms until puberty, at which point they may develop breasts and have less body hair and less muscle mass than typical XY males. They are usually infertile as well. Klinefelter syndrome is also present in other species. If you had wanted to claim P.T. Barnum's prize for a male calico cat, you could have done so if you found a Klinefelter male. Turner syndrome is called monosomy X. Turner's syndrome patients are anatomically female and they are sterile. However, they don't have any outward symptoms, which, given what we know about X chromosomes, makes sense. Any human, male or female, only requires one functional X chromosome to survive, and Turner syndrome is the only viable human monosomy. Trisomy X, or triple X, also yields an anatomical female. Again, few symptoms present in trisomy X, and women with the condition are typically fertile. With all aneuploid conditions that I've described, they tend to occur in the smallest chromosomes, or the sex chromosomes. This is because, in essence, it is the least amount of damage that non-disjunction can do and still lead to a live human birth. Other types of inborn disease caused by chromosomal problems arise when sections of chromosomes are not in their usual places. This chromosome breakage can lead to 4 types of altered chromosomes. Deletions, like the delete key on your laptop, remove material, writing on your laptop, or DNA in chromosomes. Duplication of a segment is kind of like copying and pasting a segment of a chromosome. An inversion flips the orientation of a segment of a chromosome, so it is backwards. Translocation involves the movement of chromosome segments to a different chromosome. So let's look at some illustrations. First, a deletion. For simplicity's sake, the chromosome has been labeled using just a few letters rather than hundreds or thousands of different genes. In a deletion, you can see that the D segment is gone from the lower chromosome. When a segment is duplicated and added in, you have 2 copies of this region BC. In an inversion, the segment BCD is flipped around and the resulting chromosome goes from ABCDE to ADCBE. And finally, in a translocation in which part of one chromosome can be exchanged with another chromosome. Just a couple of examples of human syndromes caused by altered chromosomes. Cri-du-chat is caused by a deletion in chromosome 5, which leads to a baby being born with severe developmental problems and a characteristic cat-like cry. One of the first types of cancer traced to a chromosomal problem was chronic myelogenous leukemia. In CML, parts of chromosome 9 and 22 translocate in cells in the bone marrow. This leads to tumor formation and this particular form of cancer. This translocated chromosome is called a Philadelphia chromosome after the city where it was first discovered. This ends chapter 15, so once again, the learning objectives, and our next chapter will take our focus from the chromosomal scale down to the DNA molecules themselves.